Hi everyone. Welcome to General Biology 1. And for today, we will be discussing the first topic for this week, which is the scientific method. Now, this is basically how the scientists study the natural world. And the idea actually of biology as a rapidly changing field may seem strange if you think of science as just, a collection of facts. After all, the parts of a frog are the same now as they were 50 or 100 years ago. But memorizing frog anatomy is not the same as thinking scientifically. Scientists use evidence to answer questions about the natural world. And if you're going to compare a frog to a snake, for instance, can you determine how the frog can live in water and on land, whereas the snake survives on desert? You see, understanding anatomy simply gives you the vocabulary you need to ask these and other interesting questions about life. Now, for this chapter, we will be discussing the different parts or different topics under the scientific method, which are the different interrelated parts of the scientific method, what is an experimental design, how are theories comprehensive explanations to different concepts, and what are the different limitations of the scientific inquiry? So, first of all, we have to ask what is a scientific method? Now, basically, scientific knowledge arises from application of the scientific method, which is a general way of using evidence to answer questions and test ideas. People are usually under the impression that science is just, it is just, a step-by-step process, and that is not at all true. Instead, science actually combines thinking, detective work, communicating with other scientists, and noticing connections between seemingly unrelated results. And the resulting insights have taught us everything we know about the natural world. Now, the power of scientific thinking is its combined emphasis on logic and creativity. And I think the best example that I can give at the moment is how the COVID-19 vaccine is being used and developed. Now, you can see here an image or like a diagram of how usually scientific method is done. And in this case, we'll be using how the coronavirus or the COVID-19 vaccine was developed. So first of all, we have to remember that it all starts with observation. So you have to make an observation. Say, for instance, we noticed that nowadays coronavirus is common and it causes serious illnesses. And we know for a fact that vaccines are often cost-effective ways to prevent such illnesses. Now, after you have made your observations, you have to ask yourself a question. Is the COVID-19 vaccine effective? Now, you have to consult prior knowledge into this until you can already formulate your own hypothesis. Say, for instance, we can say that the vaccine will stimulate the immune system and will be associated with reduced incidence of the coronavirus illness, because we know for a fact that having being vaccinated by the COVID-19 vaccine does not entirely free you from contracting the virus itself. It just, lessens the severity of the effect of the coronavirus to the person who was targeted by the virus. Now, after that, you have to make your own predictions. Say, for instance, if the vaccine is effective, then the vaccinated people should have lower incidence of COVID-19 illness than those that are receiving the placebo. And then after that, you have to collect and interpret the data, wherein the researchers measure disease incidence among people under study. And now you have to again consult prior knowledge and you have to draw conclusions. So you can say results suggest that the vaccine was safe and effective in this study. Now, it actually doesn't end in this. As you can see, this is a cycle, not just a process. So after you have drawn your conclusions based on the collected data, you have again to make an observation. So okay, so this image that you can see here is a diagram of how the scientific method is usually done. So like what I have mentioned, it all starts with, observations and questions. So you have to ask a question, You do basic background research by consulting, so you can find prior knowledge about the topic. Then you construct your hypothesis and then you test it out. If your procedure is working, then you go ahead and analyze the data and draw your conclusions. But if your procedure is not working, then you have to troubleshoot the procedure and you have to check the steps carefully on which step do you have to change something. Now, once you have already troubleshoot your procedure and you see that again, the procedure is working, then again, you analyze and you collect the data. And after that, you collect first and then you analyze the data and then you draw your conclusions. Now, if your results align with the hypothesis, then you have to communicate your results. But if the results align partially or not at all with the hypothesis, then your experimental data becomes background research for a new or future project. Now, again, you have to ask a new question and start the process all over again. So that's just how a scientific study works. Now, like what I have mentioned, all scientific method or a scientific inquiry starts with observations and questions. So the observations may, rely on what we can see, hear, touch, taste, or smell, basically what our senses give off. And it may also be based on existing knowledge and experimental results. Now, often, a great leap forward happens when one person makes mental connections between previously unrelated observations. A very good example for this is Charles Darwin. So, when he developed the idea of natural selection by combining his understanding of Earth's long history with his detailed observations of organisms. Now, another great advance occurred decades later when biologists realized that mutations in DNA generate the variation that Darwin saw but could not explain. Now, next step is the hypothesis and prediction. So, first of all, a hypothesis is a tentative explanation for one or more observations. The hypothesis is the essential unit of scientific inquiry. And for this to be useful, the hypothesis must take note, it must be testable. That is, there must be a way to collect the data that support or that can support and or reject it. Now, interestingly, no hypothesis can be proven true because, future discoveries may contradict today's results. Nevertheless, a hypothesis becomes widely accepted when multiple lines of evidence support it, no credible data refute it, and plausible alternative hypotheses have been rejected. Now, a hypothesis is a general statement that could lead to specific predictions. Now, often, a prediction is written in or as an if and then statement. As a simple example, suppose you hypothesize that your lawnmower stopped working because it ran out of gas. A reasonable explanation would be if I put the fuel in the tank, then my lawnmower would start. Next, we move on to data collection. So investigators draw conclusions based on data. So the data may come from careful observations of the natural world, an approach called discovery science. Now, a good example for this is the National Audubon Society's annual Christmas bird count. So for more than a century, thousands of citizen scientists, so we'll call them citizen scientists, have spent Christmas Day documenting the ups and downs of hundreds of bird species nationwide. Another way to gather data is to carry out an experiment to test a hypothesis under controlled conditions. Now, discovery and experimentation actually, they work hand in hand. Consider, the well-known connection between cigarettes and lung cancer. In the late 1940s, scientists show that smokers are far more likely than non-smokers to develop cancer. Since then, or since that time, countless laboratory experiments have revealed how the chemicals in tobacco damage living cells. Now, the last step whenever you do a scientific inquiry and you saw that, your procedures work, that the data you have collected somehow, makes sense, you have to analyze it and you have to send your results over for peer review. Now, after collecting and interpreting data, investigators decide whether the evidence supports or falsifies the hypothesis. And take note, often the most interesting results are those that are unexpected because they provide new observations that force scientists to rethink their hypothesis. Science advances as new information arises and explanations continue to improve. Now, once a scientist has enough evidence to support or reject hypothesis, he or she may write a paper and submit it for publication in a scientific journal, which you should look forward to because on term three, you all will be working on a research proposal. Now, um, in a process, called peer review, these scientists independently evaluate the validity of the methods, the data, and conclusions. And overall, peer review ensures that journal articles, the tangible products of the global scientific conversations, are of high quality. Now we move on to the next topic, which is the experimental design. Now, scientists test many hypotheses with the help of experiments. Now, an experiment is an investigation carried out in controlled conditions. This section considers a real study that tested the hypothesis that a new vaccine protects against a deadly virus. Okay, say for example, by the way, in this chapter or section, we will be using the rotavirus as our example. So this is actually a kind of virus that causes severe diarrhea and takes the lives of hundreds of thousands of young children each year. Okay, so first of all, when doing an experimental design, we have to consider first our sample size. Now, one of the most important decisions that an investigator makes in designing an experiment is the sample size. Now, sample size is the number of individuals assigned to each treatment. So the sample size in the rotavirus study was approximately 100 infants per treatment. So in general, we say that the larger the sample size, the more credible the results of a study. Next, we have variables. Now, a systematic consideration of variables is also important in experimental design. And variables are changeable elements of an experiment, and there are usually several types, but generally we have three. The first one is the independent variable, which is the factor that an investigator directly manipulates to determine whether it causes another variable to change. So in the rotavirus study, the independent variable was the dose of the vaccine. Next off, we have the dependent variable, which is any response that might depend on the value of the independent variable, such as the number of children with rotavirus-related illness during the study period. And then we also have what we call as the standardized variable. Usually we call this as the constant variable or the constant. This is anything that the investigator holds constant for all subjects in the experiment, ensuring that the best chance of detecting the effect of the independent variable. Because rotavirus infection is most common among very young children, the test of the new vaccine included only infants younger than 12 weeks. Now, furthermore, vaccines work best in people with healthy immune systems, so the study excluded infants who were ill or had weak immune system or immunity. Um, Age and health were therefore among the study's standardized variables or constants. Now, as you can see here, by the way, we also have what we call as controls. Now, well-designed experiments compare one or more groups undergoing treatment to a group of untreated or normal individuals. The experimental control is the untreated group, and it is important because it provides basis for comparison in measuring the effect of the independent variable. And ideally, the only difference between the control and any other experimental group is the one factor being tested. Now, experimental controls, it may take, several forms. Sometimes the control group simply receives a zero value for the independent variable. If a gardener wants to test a new fertilizer, for example, in her garden, she may give some plants a lot of fertilizers, others only a little, and still others, the control plants, none. In medical research, a control group might receive what we call as a placebo, which is an inert substance that resembles the treatment given to the experimental group. The control infants in the rotavirus study received a placebo that contained all components of the vaccine except for the active ingredient. Now, this table here summarizes the experiment in itself. So again, the sample size is 100 infants. Now, for the variables, so what they tested is the dose of the vaccine. Now, they tested it on the infants. And then the standard variable would again be the age and health of children in study. And for their control group, instead of giving them vaccine, they gave them, just a placebo, which lacks the active ingredient in the vaccine. Okay, so here is the statistical analysis of the results of the experiment that they have done in the rotavirus vaccine. So once an experiment is complete, the investigator compiles the data and decides whether the results support the hypothesis. So look at this figure here. Now, let me ask you a question. Did the vaccine prevent illness, or do the data simply reflect, random chance? Now, the researchers concluded that the vaccine was effective, but only after applying a statistical analysis. Now, researchers may use many different statistical tests depending on the type of data. You will know that when you have your statistics subject. All such tests considers both variation and sample size to yield a measure of statistical significance, which is the probability that the results arose purely by chance. Okay, so next off, we have theories as comprehensive explanations. Okay, so outside science, the word theory is often used to describe an opinion or, just a hunch. For example, immediately, after a plane crash, experts offer their theories about how or about the cause of the disaster. Now, these are just tentative explanations, which are just untested hypotheses. On the other hand, in science, the word theory has a distinct meaning. Like a hypothesis, a theory is an explanation for a natural phenomenon, but a theory is typically broader in scope than a hypothesis. Now, for example, we have the germ theory. So if you're not yet familiar with the germ theory, this is the idea that some microorganisms cause human disease. So this is the foundation for medical microbiology. Now, individual hypotheses relating to the germ theory are much narrower, such as the suggestion that rotavirus causes illness. Now, not all theories are as large as the germ theory, but they generally encompass multiple hypotheses. So, note also that the germ theory does not imply that all microbes cause disease or that all diseases have microbial causes, but it does explain many types of human disease. Now, a second difference between a hypothesis and a theory is acceptance and evidence. You see, a hypothesis is tentative, whereas theories reflect broader agreement. But this is not to imply that theories are not testable. In fact, the opposite is true. Every scientific theory is potentially falsifiable, meaning that a particular set of observations could prove the theory wrong. The germ theory remains widely accepted, not because, it is not testable or it is not falsifiable, but because many observations support it and no reliable tests have disproved it. The same is true for the theory of evolution and other scientific theories. Now, another good quality of theory is its predictive power. Now, a good theory not only ties together, many existing observations, but it also suggests predictions about phenomena that have yet to be observed. Both Charles Darwin and naturalist Alfred Russel Wallace used the theory of evolution by natural selection to predict the existence of a moth that could pollinate orchid flowers with unusually long nectar tubes. So that moth is what you can see here on the picture. Now, it's only decades later that scientists discover this long-tongued insect. Now, a theory weakens if subsequent observations do not support its predictions. Now, a good question is, what is the relationship between facts and a theory? Now, one definition of the word fact is a repeatable observation that everyone can agree on. That's how we know that something is a fact. It is a fact that a dropped pencil falls towards the ground. No reasonable person disagrees with that statement. Gravity is a fact. Gravitational theory explains the forces that cause pencils and other objects to fall. Now, as you can see here on the table, this is how we define hypothesis, or how we would know what's the difference among hypothesis, theory, and facts. Now, biologists also consider biological evolution to be a fact, yet the phrase theory of evolution persists because evolution is both a fact and a theory. Both terms apply equally well. The evidence for genetic change over time is so persuasive and comes from so many different fields of study that to deny the existence of evolution is unrealistic. Nevertheless, biologists do not understand everything about how evolution works. Many questions about life's history remain, but the debates, this just swirl around how, not whether, evolution occurred. Now, we move on to the last topic, which is the limitations of the scientific inquiry. Okay, so scientific inquiry is neither foolproof nor always easy to implement. One problem is that experimental evidence may lead to multiple interpretations, and even the most carefully designed experiment can fail to provide a definitive answer. Consider the observation that animals fed large doses of vitamin E live longer than similar animals that do not ingest the vitamin. So, does vitamin E slow aging? Possibly, but excess vitamin E also causes weight loss, and other research has connected weight loss with longevity. Now, the question is, does vitamin E extend life or does weight loss? The experiment alone does not distinguish between these possibilities. Now, another limitation is that researchers may misinterpret observations or experimental results. For example, centuries ago, scientists sterilized a bottle of broth, corked the bottle shut, and observed bacteria in the broth a few days later. They concluded that life arose directly from the broth because they assumed that by heating the broth, it should have killed all the microorganisms in the broth. The correct explanation, however, was that the cork did not keep airborne bacteria out. Although scientists may make mistakes in the short term, science is self-correcting in the long run because it remains open to new data and new interpretations. Now, a related problem is that the scientific community may be slow to accept new evidence that suggests unexpected conclusion. Every investigator should try to keep an open mind about observations, not allowing biases or expectations to cloud interpretation of the results. But, it is human nature to be cautious in accepting an observation that does not fit with what we think we know. The careful demonstration of life does not arise from broth surprised many people who believe that mice sprang from moldy grain and that flies came from rotted beef. More recently, it took many years to set aside the common belief that stress causes stomach ulcers. Today, we know that a bacterium causes most ulcers. Now, Although science is a powerful tool for answering questions about the world, it cannot answer questions of beauty, morality, ethics, or meaning of life. Nor can we directly study some phenomena that occurred long ago and left little physical evidence. Now, consider many experiments that have attempted to recreate the experimental or the chemical reactions that might have reproduced or produced life on Earth, on the early Earth. Although the experiments produce interesting results and reveal ways that these early events may have occurred, we cannot know if they accurately reflect conditions at the dawn of time. Now, we have to take this as a takeaway for this lesson that science does not promise absolute truth, nor does it consider that such a thing necessarily exists. Science does not even promise that everything in the universe is amenable to a scientific process. Now, we remember that although science is based on facts, science is not the only and absolute truth.